Okay. Okay, so our second class, we will be, first of all, briefly go over, and then we continue to talk about that. So we talk about what is microbiology. It is a science of the microorganism, and with algae, bacteria, virus, protozoan, fungi, and in the class we'll focus on the bacteria. Then we talk about what is microorganism. It is too small to see, so we need using naked eye, so we need using microscope. And this is a category of the microorganism. There is a cellular, acellular, and different category. Then we say sometimes the bacteria is always bad, microorganism is always bad because it's caused disease, and there are some of the examples. Here are some more examples, which is in the animals, plants, human beings. But don't forget, the bacteria sometimes is good, because there is doing fermentation process will generate antimicrobials, antibiotics, and some of the fermented products. And we talk about the vaccination, because recently, in recent years, we talk about the COVID-19 vaccination all the time, and you also have a flu vaccination due all the years. So we talk about the vaccination, the history, what is vaccination, and then we move on to the discovery of the microorganism, a little bit of history. Like Anthony van Leeuwenhoek is the first person using his mechanic microscope to do the observation of microorganism and clearly, accurately recorded. Then we talk about Louis Pasteur, his swan neck flask in a microbiology level, repel spontaneous generation theory. And then we talk about John Tidal and Ferdinand Cohn, both which is finally tells us why there is some of the contradictory during the Louis Pasteur's research because of the heat-resistant microorganism and the endospore-forming microorganism. So today we're going to move on to the next couple of things, which is very important for the history, and we will talk about the real bacteria. So we may bring some of the demo here, okay? So let's talk about Bob Koch. Bob Koch, I want to make sure the spelling is correct. Bob Koch, K-O-C-H. Bob Koch is a great German microbiologist. He did, I will say he's the founding father of modern microbiology. Typically, for one disease called Mycobacterial tuberculosis, and he's the first person who is well established a relationship of a microorganism and their caused disease, which is a very important and the famous theory called Koch postulate. This is described about the relationship between a microorganism and a disease. Now, obviously, this disease is we call it an infectious disease. So, what is his postulate? Number one, I would say very simple, healthy biological body should be absent from disease-causing, I will say, disease-causing microorganism. So I give you a very easy example. Here is Mycobacterial tuberculosis. Mycobacterial tuberculosis. This is acronym, just call it TB. and you know that it's a very severe lung disease. And I want to tell you one thing. 100 years ago, or 150 years ago, before antibiotics comes out, if you get microbacterial tuberculosis, you have TB, basically means you died. There is no way to do any of the chemical treatments to do it. And I was originally from China. In the Asia and China, they try those traditional herbs and the medicines is not going to work. Why? We will mention later on. This bacteria has a mycolic acids in cell wall. What this means, this cell wall is very waxy. You know what the waxy means? In the old time, if you have a dorm, you have a floor, you don't want to clean all the time, you put a wax in. So very sleepy, very sliding, very slippery, we say it. What this means? Because it's basically is a different combination of the chemicals. So if the medicine is based on water solution, it's not going to penetrate it. Let's say you have like herbs, antibacterial herbs. You dissolve into water, then you drink it. It's not going to penetrate the cell wall, so it's not going to affect it at all. Until 1950s, 1960s, the antibiotics has been generated. Then they conquered this guy. And even today, still lots of places have tuberculosis. 150 years ago, when you have this, you almost means you'll die. It's like a lung cancer by the time. So that's why Bob Cook have to do the research. The target is microbacterial tuberculosis. So let's say a person who has TB obviously means he will have microbacterial tuberculosis in his body. If a healthy people, he or she should not have microbacterial tuberculosis. That's the first item. Okay, so the second one. Now, obviously, this means the sick people will be contaminated, we can say, okay, contaminated with the target microorganism. The second one, this target microorganism can be isolated, and very important to get pure culture. And we will mention this real quick. What means pure culture? What this means, if people who has TB, microbacterial tuberculosis, you could get a pure culture of microbacterial tuberculosis from this patient. Let's say from sputum sample, from lung area, and they can grow in a pure culture. Okay, then the third one. The pure culture infected a healthy person will get similar or same disease, which means you get isolated this microbacterial tuberculosis, you go to infected a healthy people's lung area, or even going through the bloodstream, you're going to get same TB. Similar symptoms. The last points of his postulate, and the same microorganism can be re-isolated from the target patient. Which means These healthy people infected with microbacterial tuberculosis have similar symptoms, the same symptoms. So I get a sputum sample from him or her, we could get isolated of the same microorganisms. That's why we call it re-isolation. And obviously Bob Cook doesn't do this in a human being. He's using guinea pig as an example. What kind of guinea pig? Looks like a mice. He used guinea pig as a model to do the test. And it comes out this very famous and important Koch postulate. But I want to mention one point here. Is what do you think is the first one? The healthy biological body should be absent from disease-causing microorganisms. The sick person will be contaminated with. Does this always to be true? Not really. They didn't consider about asymptomatic person. Is that right? People could be carrying on the pathogen but never show any symptoms. So I gave you an example like Magic Johnson, who has a carrier of HIV virus. And the people even scared to play with him in the NBA league matches. But he never have a problem by himself. Now what that means, which means it's a little bit the story of the immunology. But you have to know, when Bob Koch mentioned this, it's somehow 80s, 70s. At that time, there is no immunology comes out. Remember when the immunology will be developed after DNA and RNA, when the DNA comes out, 1953, Crick and Watson mentioned about the double helices. That's almost 70, 80 years later. So by the time when he mentioned this Koch postulate, there is no theory about immunology yet. So that's why the first item is questionable right now, however, it's still important by the time. Okay? So something I want to mention. Okay, next one. We talked about this pure culture. So we want to mention to you and introduce to you how we get isolated of the pure culture, of the pure culture. In the history, there is a two group who are the competitor. I mentioned Louis Pasteur and Bob Koch. And do you think they're friends? No, not really. They're competitor. One from France, one from Germany. They're competitor. They're great microbiologists, but all the time they're competitor. By the time they are competing to get pure culture. This is very difficult to get, I tell you. It's a magic. You cannot even imagine right now, it's still a magic stuff. Louis Pasteur's group, think about, we could get pure culture from liquid solution. Now can we do that? We could try. Okay, I had a bacteria solution right here. Let's say E. coli. I grow 35 degrees Celsius 24 hours. It becomes very turbidity, cloudy. So the bacteria is there. You know, you see any of the solution very cloudy or turbidity, that means bacteria is there. So what we do? Let's do serial dilution. Let's do 10 or 100-fold serial dilution. How to do it? You did it in your biochemistry or chemistry lab. Some of you, how we do? We prepare these 9.0 ml nutrient broth. We're going to prepare a lot. Okay, we prepare a lot. Let's say we prepare 5. So we're going to add 1.0 ml onto the first one. So we mix very well, then add 1.0 ml to the second one, mix very well. Then go add 1.0 ml, go to the third one, mix very well. Then add 1.0 ml, go to the fourth one, on and on, on and on. So finally, we assuming there is one cell in the tube solution. Can you do that? Theoretically, yes. Practically, no. Because you can verify there are one cell there. And you can verify these are only E. coli and one cell E. coli. Is that right? So how we should do it? Then Bob Koch said, okay, that's not a good idea. We have to do this on solid media. But then give you a question. If it's a solid media, what type of the material we'll be using? That's a question. So why it is difficult to do? The material, bacteria, has to grow. This is number one. Number two, bacteria cannot eat it, which means they cannot block it. If they block it, you don't know where is the bacteria going to go. And also, this has to be solidified at room temperature and have to be melted at high temperature. This material is very tough to get. So they tried a lot of material. At the beginning, they tried jelly. What kind of the jelly? Grape jelly. Looks like the one put in the middle of your sandwich, on your breakfast sandwich. Put a jelly. Can the bacteria grow there? Yes, but they're going to block it. So you're not going to see it. So what they do? It's very interesting, I'll tell you one thing. There is one person work in Bob Koch's lab, is called Fanny Hesse. It's not really Fanny Hesse, it's actually his husband is Walter Hesse. So Walter Hesse got a job from Bob Koch. He said, go ahead to find the solid material to isolate the pure culture of bacteria. One is he tried lots of material and they failed all the time. This is what the science they do. They fail all the time. At the end of the day, they get successful. Then a very hot summer, 80s, hot summer in Germany. In Germany, countryside of Germany. Okay, very hot. There's no air conditioning by the time. Walter Hesse's wife, Fanny Hesse. He said, honey, the lunch is ready. Can we have lunch? And I prepared today a very special French pudding. That's what you like it. A French pudding. Okay, then what has he thought about pudding? That's really good. What's a pudding comes out? Polysaccharides. It's a basement of the agar. So then he go back to the lab and he tried, and it's successful. This is what it looks like today. A bacterial solid media looks like an agar. And we can streak bacterial solutions there, and we can do the incubation. And what we gonna got is a bacteria colony. Looks like this. There is no lab for this class, that's why I have to show you. That's each individual colony is a pure culture. But this is not the real message to get it, I just to show you. Okay, how we should get a message is right here. If we get a bacteria place, this is our agar place. Okay, how we can get a bacteria pure culture? This is a bacterial solution right here. 35 degrees Celsius, 24-hour, E. coli solution, very crowded, all the bacteria there. You need to use a plastic loop, do a very intensive streak at the beginning. We call, this is called number 1. Then you have to flame your loop. And on the side of the first course, do a less intensive second streak. This is, we call it a second course. And then what you do, from the side of the second streak, do a third one, less intensive. Then you do a last one. And this other plates, again put at 35 degrees Celsius, 24-hour. What you will see, those very tiny dots there. This is called a pure culture. What is a pure culture? A group of bacteria cells originated from a single mother cell representing one gene name and one species name. That's what's a single cell for. So, for example, what I have is Salmonella typhimurium here. This single colony, which is indicate a pure culture, it is only one genome name is Salmonella, and one subspecies name is typhimurium. So I will explain to you real quick. Salmonella is a little bit tricky, different to E. coli. Okay? So let's back to talk about history. Fanny Hesse found it. The agar, which is a polysaccharide from seaweed, is a good material, a solid material to grow bacteria, can get isolation of a pure culture. So where should be the container looks like? Another great scientist from Koch's lab find it. His name is Richard Petrie. And he find this kind of the container could be, hold on, melted agar. Agar will be melted at 55 degrees Celsius and solidify at room temperature 25 degrees Celsius. It looks like this. It's a round shape. By the old time, it is rectangle. Like this. Right now is round shape because you know the same perimeter circle have a big-ish area. Is that right? That's a simple math. That's why this type of the material today we call it Petri dish. This is used to memorization. A great microbiologist, Richard Petrie, who is the first one created this type of the material as a container of a bacteria agar and a microorganism agar. It's a very magic stuff. Today, we do this every day in the life, in the lab. Petri dish, a melded agar, and a growing bacteria on the agar. It's very simple, looks like a routine job. I did this for 20 years, since I was a student like you. But 150 years ago, it's a breakthrough of modern microbiology area. Another thing I also want to mention, very important you need to know. What I'm talking about here is a pure culture. It is a group of bacterial cells. This is not a single cell. This is very tricky there. This colony, we call it a bacterial colony. How many cells are there? Around 10 to the 6 cells. That's why we say a group of bacterial cells. It's not a single cell. It is different. Okay? But they are all a same genus name and same species name. They are same E. coli, they are same Salmonella, they are same Bacillus. They should not be contaminated with other bacterial culture. Okay? So that's important for the pure culture. Okay, so that's Fanny Hesse, and this is a pure culture developed using agar and isolated from seaweed, and the complex of polysaccharides. Most of the bacteria could use it to remain solid, even a wide range of the temperature. Just wanted for you to recall, we mentioned, but I want to let you know, 50 to 55 degrees Celsius, the agar will be melted. Okay, very sticky. And the room temperature usually 35 and 25 degrees Celsius. This agar will be solidified. Solidified. Like what I have here. Okay, so that's very simple. And this is a strict flaking. We get the pure culture. That's a colony there. The isolated pure culture. And we all know that, same like other biological body, bacteria use binomial nomenclature, has a species name and genus name. So, but something I want to emphasize to you, a little bit of tricky thing will happen. So, what the story is? Let me move this out. Even people got PhD, called master degree, doing microbiology for many years, they are still made a mistake lots of the time for this enumeration system. Okay. Binomial nomenclature. Okay. Which means two names, one's genus name, one species name. Okay, if it's E. coli, what is the full name of E. coli? It's Escherichia coli. You have to write it italic. The first name is Escherichia, genus name. Most of the time, this is named by a person, to be honest. For example, Escherichia coli. Escherichia is from a microbiologist from Japan. Okay, sometimes it's a person. Coli is a morphology actually, but it's a species name. Species name. But there's one exception. Okay, I can write more here. Bacillus subtilis. Listeria monocytogenes. That's all the same. The first genus name, the second species name, binomial nomenclature. But what happened for one exception? The exception is Salmonella. The correct way to write a Salmonella, let's say Salmonella Newport should be Salmonella. This is italic. Then the Newport is normal. Non-italic. Why? Because the full name of Salmonella Newport is Salmonella enterica subspecies enterica. Bacterica Newport. All these from here are italic. The last one is normal, non-italic. However, we think this name is too long. We took this out and we combine them. So, a simple question for you. Why Salmonella Newport? The Newport is non-italic. Because this is subspecies name, not species name. Very simple. It's an exception. Okay? So once you read a paper, read a book, if you see Salmonella Newport, the Newport is still italic. Even sometimes Salmonella typhimurium, the typhimurium is still italic. That's wrong. That's not very correct. But people are doing those things all the time. Okay? Here, the slide was showing you there's a no exception. Okay, you see Bacillus arthritis. Bacillus is genus name, arthritis species name. That obviously is a pathogen causing arthritis. Clostridium botulinum, we're going to mention Clostridium species name. As a genus name, botulinum, genus name. E. coli, Escherichia coli. Streptococcus pyogenes, genus name and species name. Streptococcus pyogenes will cause strep throat. And this is a pathogen actually cause a strep throat right there. The Streptococcus pyogenes. Okay, we will mention detail in the exam 3, we talk about that. Okay, so this first one we finished. This is gave you an introduction what we have for the microbiology and a little bit of history. And the next one we will spending today and also next week Tuesday we're going to talk about microbiology. And we will go very detailed, talk about bright field microbiology, and then we will talk about dark field microbiology, microscope, laser confocal microscope, electron-analyzed microscope, and a little bit something else. And we will have a lab section later on to talk detail about how we're going to use it. Okay? So, let's talk about microscope. We already said that the microorganism is too small to see. So we need a microscope. Microscopy, we also call it. What type of the microscopy we have? We listed there. Basically, we have light microscope and electron microscope. What's the difference between that? The major difference is energy resource. Light microscope using light as an energy resource, electron microscope using electron beam. And we will talk about that. What are the light microscopes? The example. Bright field microscope. Dark field microscope. Phase contrast microscope. Fluorescence microscope. And laser confocal microscope. What are the electron microscope? Scanning electron microscope and transmission electron microscope. Acronym is AEM, and TEM. Our university, in the plant science building, another class what I'm teaching is a major microbiology class, which is AEM 341. We had a lab section. We had bright field microscope. And we used to have dark field microscope and a phase contrast. I'm not sure they're still working right now. Laser confocal microscope is a very expensive equipment. That is almost $50,000 above, north $50,000. Scanning electron microscope, transmission electron microscope at our engineering building at WVU. They have a group, the shared facility, they have it. If you want to use it, you need to get a training to do it. So just give you a brief view of what we have. So our class will be focused more detail, talk about bright field microscope. The structure, what it looks like, and what's the story behind it. First thing, this is a little bit mass, and you should know. Okay, 1 centimeter, 1 millimeter, 1 micrometer, 1 nanometer, and 1 angstrom. I don't think we do angstrom anymore. So 1 nanometer is good. That's a calculation. How about that? Light going through a different media will have bending, refraction. Going through lens will happen is a confocal, focus, and then we have a working distance, all these things. We're going to explain to you one by one, okay? So, first of all, the first terminology we want to mention for a microscope. This is a bright field microscope, very similar to the one what we use in the lab. So the first question I always ask all my students, this microscope pretty much somehow $2,000, $5,000, depending on the different resources. What is the most expensive part of that microscope? What do you think? It's a lens. Yes, the lens is the most expensive part. Because the lens will do a good job of magnification. This comes out with our first question. What is a total magnification of a bright field microscope? This is calculated by total magnification equals magnification of ocular lens multiplied by objective lens. Where is the ocular lens? It's always the one on the top. So your eye can go there. So that's why we say it's ocular. What's the objective lens? The lens at the middle, at the bottom. Okay? So let's do a very simple calculation. If the ocular lens, it is always 10x. And the objective lens, we have different type. If it is also 10x, what's the total magnification? It's equals 10 multiplied by 10 is 100x. If the objective lens is 40x, what's the total magnification? 10 multiplied by 40 equals 400x. If the objective lens is 100x, this is the one we always use in the lab. 10 multiplied by 100 equals 1000x. That's a total magnification. Now, what's the lens looks like? The lens we can draw briefly looks like this. Second one like that. Okay, then the last one like this. So third one like this. The fourth one like that. There's a stages right here. That is stage. So it is 4x, 10x, 40x, and 100x. This comes out with our second working distance. What is the working distance? It's a distance of the lens to the stage. Can you see it? It is decreased with the increasing of the magnification. Can you see it? Obviously, it is decreased with increasing of magnification. Then we'll have one problem. Okay, this is light. The light is right here. Okay, the light is right here. When you turn on the button, the light is on. So the light going through the water. What will happen? It's very interesting. You will see like this. When the light goes like this. The light goes like this. Goes like that. Goes like this. Like that. Like this. Like this. And goes like this. Goes like that. Like that. What I call refraction. Because the light goes through different media. This is air. This is glass. So what happens is refraction. What means refraction? When you see it, you're feeling it's fuzzy. You're not going to see it very clearly. So let's say we in the lab, you always use 10x to find the specimen. Then we transfer to 100x. You will find it's not even as clear as 10x. Very fuzzy. Because of refraction. Okay, then what are we going to do then? It's a very magic way to do it. Called oil immersion. This is really amazing stuff people find at the beginning. How they do it? They put one drop of a big oil here. And then you focusing. You will find a very clear picture. No problem. Why? Because the refraction index of air equals 1.0. The refraction index of glass is 1.5. And the refraction index of oil also equals 1.5. Therefore, when you put oil there, you kind of make oil and glass or a whole system. So what's the oil immersion for? Remove air gap. That's why in our lab practice, we are doing the observation of the bacteria. We always use 10x to find the sample. And then we put a drop of the oil, a big drop at the middle. Then we switch to 100x. But there is another question. Okay, I'm spending a lot of time to find the 10x, find the specimen. Then I put a drop of the oil, I switch to 100x. I may lose focus. Is that right? Lots of people switch to doing this, doing that, put the fine focus there, or cross focus. And I want to tell you one thing. These type of the bright field microscope, they don't have this problem. Why? Because they have a very good technique, a good characteristics of that is called a parfocal. Which means once focused, lens stay focused. This means if you're using 10x to find a specimen, and you transfer to 100x with oil immersion, only need minimal adjustment. You will get the job done. So nice it is. Well, technically in the lab, you will try later. It's not work all the time. The reason is those lens is not very clear, and the fine focus or coarse focus sometimes loosened. But if I brand new bright field microscope, it has a very good technique. It's called a parafocal. Okay, that's very nice. Last question, what we want to solve. Resolution power. So why we use microscope? Go back to this old question. We want to use microscope because we want to see bacteria, and the bacteria is too small to see using naked eye, which means the resolution power is not good enough for our naked eye, which means the ability to differentiate two closely attached particles is not good enough. So we need to use microscope. Every microscope have a different resolution power, and there is a basic formula can do the calculation. RP equals lambda divided by 2NA. Okay, what is that? This is a formula equation. Originally, this is coming from the triangle formula. It's a little bit complicated, but we're not going to go through those detailed math. Just very basically to talk about that. What is lambda? The bright field microscope, what we are using in the lab, like this, a 5,000 one or 2,000 was a cheaper one. When you turn it on, and then you switch it all the way to the end, the lambda is always 625 nanometers. It's a fixed number.